Hey, welcome to the Neurophysiology whiteboard session. It's going to be section 4. We're going to be discussing action potentials and synapses. So let's get started here. First, we're talking about an action potential. Let's get our pen. Okay. All right. First, we're talking about action potentials. Okay. Action potentials, AP. All right, so first, I would say that an action potential is going to be an electrical current that is generated in one location on an excitable membrane. Okay, so if we have a membrane here, okay, let's do a membrane. So an action potential is basically just going to be a sort of singular event, okay, that is happening in one location, and it is generating a electrical current, okay. And this is on a is referencing a excitable membrane, okay, and we'll talk a little bit more about that. So once this action potential, okay, so this electrical event, right? Once this electrical event is initiated, it basically transmits across the entire membrane, okay. And one last thing that we'll say is that that an action potential is also a all or none event. Okay, so this is sort of a principle that that if the membrane is sort of like, let's just say it's in a resting state, okay, it is not undergoing this electrical current that is being transmitted across, is not in the middle of this event, it is going to be in a resting state. But then once this action potential comes in, right, this electrical current is transmitted across the membrane. Once that happens, it's done. It's all in. Okay, so there's really like no going back. Okay, so it's as if you were pulling the trigger on a gun. Okay, so I think I've read some text, a lot of texts that have used that example that you can squeeze the trigger, squeeze the trigger, but once the trigger has been depressed enough to instigate the firing, then that gun is going to fire, right? And then that is also an event that is, and this is sort of relevant here in the future, but this is an event that is reproducible and it is the same every single time. Okay, so we'll just sort of leave it at leave the basic definition of what an action potential is at that. And we need to talk about, so in 4.1 on the document, we're talking about membrane potential. Okay, so membrane potential, let's use this. Let's use this as an example over here. Give me just a second, get rid of this. Okay, let's get rid of the whole thing. We'll do a new one. And okay. So we'll say that we have this membrane, right? And so membrane, and membranes have potential of energy, okay? So the potential energy, okay, is basically going to be determined by something we call polar charge. Polar charge. Okay, and what I mean that is that within our cells, within our body, we have these membranes separating cells. We have two different compartments, okay, And if there is an overall net electrical charge that is going to be, we'll say, positive out here and then negative in here, two different compartments. And so say that this is our extracellular fluid, and then this is the cytosol within a cell, okay, separated by a membrane. The difference of the positive and negative charge that is found within these compartments has a potential of energy between that positive and negative charge, okay? And we'll also say that we can sort of couple these charges to ions, okay? So we have ions, so we say we have like sodium, right? So sodium has a positive charge, but then we have chloride, right? Chloride has a negative charge. So the difference of the concentrations of chloride and sodium, right? The positive and the negative, these have values, right? That correlate with an electrical charge. That difference in these charges are polar, okay? But they are potential energy. And if it's potential energy is something that is technically that is stored, so it's not being used yet. So if we have this membrane, this excitable membrane that is between these two polar charges, and let's say it's fairly significant, right? Let's say there's a large difference between these numbers, then we have a lot of potential energy within this, within the gradient between these two, okay? And I would also then say that if you're just looking at sort of a generic cell, okay, if you're looking at a generic cell here, and within the body, I would say that you have a higher potassium concentration, okay, concentration of potassium inside the cell, and then you have a higher concentration of sodium outside the cell. So this is the extracellular fluid, and then this is the cytosol, okay? And this is sort of a truth you need to just memorize and understand as we move forward and learn about an action potential, okay? And I would also say that a couple other things here. So you have, if we look at this sort of like generic picture of a cell membrane with positives and negatives, right? I would go on say that that this isn't actually an accurate representation of that. I would say that to be more accurate here, let's keep this out here. I would say that we have sodium and chloride, okay, concentrations in the extracellular fluid, it's more common. And then in the cytosol, okay, we have proteins. Proteins generally have a negative charge to them, and we have a higher concentration of sodium ions in the cytosol or intracellular. Okay, so let's say this general rule is that extracellular fluid has a higher concentration of sodium ions and chloride ions, which are positive and negative, and then within the intracellularly or within the cytosol, you have a higher concentration of potassium, which is a positive ion, and negative ions through proteins. So I'd say that this is more of an accurate statement there. Next, we talked a little bit already about this sort of gradient between these polar charges, right? We first need to, or we can sort of expand on that, right? And we can say that that this concentration gradient, concentration of these or the gradient of charges between these different concentration of ions, they're sort of formed in two different ways. And we have what's called active transport versus passive transport. Okay. And in active transport, we have something very, very important channel here, okay, is what we call a sodium potassium ATP pump, okay. And then we have like individual channels. And for example, this would be like a sodium or a potassium channel, okay. And you look at the words here, okay, so if you have active transport, so we're talking about transport of these ions across this membrane. So when you have an active transport, you're having to use energy. And ATP, adenosine triphosphate, is a high energy molecule in our body that we use. So whenever there's ATP involved in some sort of transport, like across the cell membrane, then you know that this is active, okay, because it requires an energy input for it to be able to perform its function. And so in a sodium potassium pump, okay, so if you have the cell and you have the sodium potassium pump, so it's a pump, it's located on the cellular membrane here, and you're essentially going to be taking some of the potassium, okay, that is intracellular, and you're going to be pumping it out. And at the same time, you're going to be taking some of the sodium that is extracellular, okay, and you're going to be taking it into the cell, okay. So you're going to be exchanging potassium outwards and then sodium inwards. They're both positive charges, okay. And I would say that the sodium potassium ATP pumps also work with a specific ratio. So for every, and this is a high yield topic, so this is very common in questions. You'll be at, like, it'll just ask, what is the ratio of sodium coming into the potassium going out, and we have 3 sodium going in to every 2 potassium that are coming out. So the sodium potassium pump has a 3 to 2 ratio, okay. 3 sodium out, 2 potassium, 3 sodium in, 2 potassium out, okay. That's a high yield point to remember, okay. And then if active is requiring ATP, then we have something that's passive, so it's just sort of happening freely on its own. And we have these channels, okay, and they're sort of located all over the surface of a cell, right? They just everywhere within the cellular membrane, okay. And these channels are passive. We call them leaky channels, okay, because they're pretty much always open and allowing for their respective ions. So you would have, maybe this would be a potassium leaky channel, and then this one would be a sodium leaky channel. So these would be just always allowing the passing, okay, of sodium and potassium, their respective ions, in and out of the cell at all times, okay. And so this is sort of allowing a constant influx and efflux of small ions and small electrical gradients that are coupled with their concentration, okay. And we'll talk a little bit more about that. That's called an electrochemical gradient. I'm going to, I guess, tell you now. So it's basically a concentration, right? So if you have a positive, if you have a low concentration of potassium, okay, versus a high concentration of potassium. So if each one of these individual potassium has a electrical has a summation of electrical charge to it, okay, then the more of that concentration that you have, right? And we already talked a little bit about this before, but the more concentration you have of this particular positive ion, then on this side of the membrane would just be more and more and more concentrated. It would also have more and more and more potential energy, right? So the electro, right? So the electric concentrate is related to concentration. Okay, so these two are related together, and that's electrochemical energy. And then it is sort of the difference of the charges that we have. So if it's very positive here and negative here, then we would then say that it is polar, and there's a gradient, right? So a gradient would be like on a hill. So if you have a hill here, right, and then you would measure if this is maybe 5 feet high, right? Then this is sort of sea level here, ground level. The difference of the 5 foot to the ground level here, this difference here is going to be that gradient. So we're saying that we have electric charge within each of the ions, and that each one of these ions have a specific concentration to it, which will add to its electric gradient across the membrane. So that's what we mean by electrochemical gradient, and that's sort of how they work with all these different types of channels, whether it being active requiring ATP or it being passive, sort of just free flowing on its own. And next, we're going to talk about sort of the basic action potential. Okay. Let me get another pen here. Okay. So basic action potential. Let's see on this side we have, let's start here. So we have 0 millivolts, we have minus 55 millivolts, minus 70 millivolts, and then we'll say plus 30. Okay, so we have a positive and a negative. Okay, so we're negative millivolts, positive millivolts. All right. What I'll say is that there is a cell, okay, that is at what we call resting membrane charge, right? So if a resting membrane of a cell is at about -70, and this is different from one text to the next, so a slight difference, but about -70 is where your resting membrane potential is, okay? And then what happens is that we have some events that are taking place that are altering the charge of that membrane, okay, and the difference of charges across that membrane, right? So we just discussed about all that, and some of those would be like the change of sodium and potassium gradient, right? So if we're at -70 at the membrane, the resting membrane, so we'll call this resting membrane charge, okay? If we start to have a more influx of positive ions inside of the cell, then what's going to happen is we're going to start to go more positive, right? Adding more positives to it, right, will make us more positive. So instead of -70, we now up to -55, right? And what happens then is that if we hit this, let's say this -55 point right here, we have, let me get this out of my way. we have an event that's triggered, okay? And sort of the event is going to be, this is what we call the threshold. So there is a threshold point, which once we get to negative 55, we can say that if we hit that, we can trigger an action potential. And sort of what we discussed before about like firing a gun, it's an all or none principle, right? So once we hit this point right here, this threshold charge, then we kick off an action potential, okay? And there's multiple phases of an action potential. So then what happens is it goes, the charge goes from that negative 55 all the way up to this point here, which is a positive 30, okay? And this we would say is going to be our peak, okay? And then after we hit positive 30, the cell is going to start dropping back down in charge again, okay? And back down in charge, and it'll actually even go further down, further negative than what it was before at resting membrane potential, before it returns back to the negative 70, okay, back to the negative 70. And what happens here is in this state right here, okay, this negative 70, right? This resting membrane potential. This is sort of maintained with those leaky channels, right? That's sort of your passive, right? Then we have that active, right? And that was like our ATP driven sodium potassium pumps, right? So we have these sort of different channels that are working, bringing in positive and negative charges within the cell, or exchanging them out of the cell back into the cell. And you're going to be getting, around this period, you're going to be getting sort of slight changes in that resting membrane potential. And if you bring in more positive, then you said we'll start to go more positive. And then once we hit this threshold, we're off, right? So that's what we were talking about previously. Now, this is sort of happening all the time. Well, I'm trying to get rid of this. It's here. Might not be able to. We'll just ignore it. We'll leave it there. So what's happening here in this phase where we're in red after we hit the threshold stimulus, all the way up to our peak, okay? This is what we call depolarization. Okay, so basically everything on this side here, okay, is depolarization, because we talked about how you have a cell, you have a positive to negative, right? So you have a positive to a negative, and you have a difference between that. That's a polarization of charges. Well, we are decoupling that polar difference, right? That gradient. So we are decoupling it. We are depolarizing. So we're going from polar, which is positive and negative, and we're bringing in a bunch of positive ions into the cell now, and we're making the inside of that cell more positive and more positive until we hit this point here, which is the negative 30. And the way I draw this, and I think that it's a good way to do this to help, you know, keep this sort of in your brain and be able to remember, is that I always just write sodium. Okay, so in the depolarization of a cell, we have sodium going in. Okay, so what's happening here is that on the cell membrane, right? So we have these sodium channels, and what's happening is the sodium is pouring in, right? And it has, so we'll say if we have this this single protein here, or this single channel here, it has some gates on it, okay? And what's happened is that these gates have been activated, all right? And they're allowing sodium to go through it, okay? But they're going into the cell. And that's exactly what these are. So these are what we call activation gates right here. So we have activation gates, okay? Then so we'll talk a little bit more about those again. But next, once we've gone to the peak here, we've fully depolarized the cell. What's going to happen then is we're going to take our potassium, okay, within the cell, and the potassium is going to start exiting the cell. Okay, so it's leaving the cytosol, going to the extracellular fluid, and sodium is a potassium ion, right? So if we're dumping more positive, right, then our overall charge is going to start going back towards the negative side. So so on the depolarization side, we were bringing in more sodium, which is positive, but on the what we call the repolarization, we're repolarizing now. On the repolarization side, we're still getting rid of positive ions to make our cells more negative, but it's going to be potassium instead of sodium. And so this continues to happen, and once potassium is starting to exit, we'll say that the inactivation gate, okay, so the gates on these sodium channels are sort of closed off here. Okay, so they're closed. All these sodium channels are closing off, and now we only have active open sodium channels. Okay, so here we go. So these are open now, right? And so now you have the leaving of sodium, and this happens till it gets back down to where our resting membrane potential is, right, which is -70. And what's going to happen though is that we're going to just keep dumping potassium out, okay, and we're going to go even more negative than what our resting membrane potential is, and that's this area right here, and this area right here is or the fact that we've gone down to maybe -80, right? Maybe -80, -85, okay, -85 millivolts. Once we've gone down to this point, we're now saying that we have gotten hyper polarized. Okay, so hyper is an increased, right? So first you decouple the charge, that polar charge, so we depolarize it. We get rid of the polarization. We go from negative and positive to just positive. So now we need to repolarize the cell. So now we need to get rid of that positive, which is potassium instead of sodium. And we get rid of so much of it, we get more polarized than we were before we even started this, and we're then what we call hyperpolarized. And I can say also that if you are in this threshold point right here, okay, till the point that you get back to your resting membrane potential, if you're anywhere in this phase of the action potential, you are the cell, we will say, is in refractory period. Okay, and this is a testable concept here, and so it's basically saying that the refractory period is the point from which you've initiated a single action potential, okay, and you are in the process of action potential, okay, and then this point here where you're back at resting membrane potential is the point where that singular event that we discussed, that single firing of the trigger, is now complete and the cell is back to a resting state. So what we can say then is that the refractory period is over, and then the cell could initiate another action potential. Okay. And while it's in the refractory period, it cannot. It is in the process of doing a singular event. You cannot do 2, 3, 4, 5, 6, 7, or however many you want all at the same time. Can't overload the cell like that. It is going to be a singular event. It's sort of all, it goes with that all or none principle that we discussed before. So in a refractory period, you cannot initiate another action potential at that time. We have a couple different kinds of action potentials, okay, and we can talk about. So we'll just try to leave all this stuff up here now. So we could talk about a chemically. So we have a chemically gated, or so let's say gated. We can also call this a ligand, Ligand, ligand gated ion channel. Okay, so we have one here. And a ligand gated ion channel is something that specifically binds to a chemical. So an example of this would be like a neurotransmitter like acetylcholine. We'll talk more about that coming up. These are also called nicotinic. So it'd be acetylcholine that binds to a nicotinic receptor, or also one that you could find on nicotinic receptor that we could find on the neuromuscular junction. Okay, and these basically are binding specific chemicals. And another kind of gated ion channel, okay, is going to be something we call the voltage gated ion channel. And these open and close in response to changes in the membrane potential. Okay, so an example of this would be like sodium, potassium, or calcium channels. Here's a good example. We have the sodium and potassium gated channels that are opening and closing, right? They have activation and inactivation gates. They're opening and closing throughout the action potential, okay? And we can also say, so we really just described all of these right here already, okay? But it's based off of voltage, right? Off the electrical gradient. And we could also then say too that that you can pretty much find these. So this is like a neuron, right? And we have our axonal processes here. This is our axon. We can say that we can find sodium and potassium voltage gated ion channels here along the axon, most abundantly across the axon. Okay, so across this point to the axon hillock right here at this point. Okay, so sodium and potassium along the axon. Then we'll say that we will most commonly find like calcium. So we'll find like calcium channels along this area here. This is sort of the end. We've discussed this in previous whiteboard session here. This is the axon terminal. Okay, so axon terminal. We'll find calcium voltage gated ion channels, and then we'll say that we'll just leave it there. So there's some common places that you'll find it. Okay, and next we have a mechanical. Okay, so we have a mechanical gated. Okay, And this is something that is stimulated by touch or something physical, right? So something tactile. And some example of these would be something that you would see in the skin. Okay, you would have like a free nerve ending, right? So this is, um, um, or we'll say not a free nerve ending. There's more pain and temperature, right? So it says not the best. So we would maybe say like the Meissner corpuscle. So Meissner corpuscle. So this is sensing touch, okay, particularly fine touch and vibration as well. So that would be something that would stimulate a gated ion channel, okay. Gated or ligon is the chemical. So these three are what we refer to as gated ion channels. Okay. Next, we'll talk about 4.4 here. Okay, so 4.4, talk about graded potentials. So let's get rid of all of this. Get a clean board. And start with a new pen. Next, we're at 4.4. These are graded potentials. Okay. And I think to sort of explain this, get this sort of generic neuron, right? With an axon, then it's an axon terminal down here, okay? So here's our nucleus. So this is sort of a generic neuron, okay? And I would say that a graded potential, okay, is, if we were just to find it, we would say that these are like changes in membrane potential, okay, that can not, emphasize, cannot basically spread from a single site or location across the cell. And well, you would say that it can't spread far. Okay, so it won't be able to spread far. Okay, and let's look at that. So if we have in this generic neuron, we have electrical stimulus is coming in and hitting the cell body, hitting dendrites, or let's say we have another neuron over here, and it has an axon coming down to hit the cell body, right? Well, it's going to be transmitting an electrical current, right? And it's going to be changing the membrane potential in this location, okay? But we're saying what a graded potential is that it's a change in membrane potential, but it can't go very far, right? So it's not enough to be able to carry this electrical charge to that point that we call the axon hillock, which is where we sort of kick off an action potential down the axon, right? Instead, it's going to sort of like diffuse out. Okay, so that's pretty much what a graded graded potential is. So the way I look at that is that it's a small potential of energy. It's being transmitted across the membrane. Okay, but so it is like an action potential, but it's just not enough to form a full action potential. Okay, and to sort of hammer that back in this in a little bit, or give a description of it. So we go back to that 0 minus 55 minus 70, right? So we talked about how we're at resting membrane potential, and then if we hit negative 55, we can go and have a full-blown action potential, right? So we just talked about all that. This is that threshold point right here. And if we look at the difference of negative 55 and negative 70, or our threshold versus our resting membrane potential, that's equal to, we'll say 15 millivolts. Okay, so this is 15 millivolts. And so the way I see a graded potential, right, is it's a tiny little action potential, or not full-blown action, but a tiny potential of energy that is being transmitted across the cell membrane. But let's just maybe say it is, let's just say that it's maybe like 0.5 millivolts, right? So if you hit 0.5 millivolts, it's not going to be enough to take it from negative 70 to negative 55. It would be negative 69 and a half, right? So that's how I see a graded potential, okay? And thing that is sort of key about this too, okay, is that these graded potentials are not formed by leaky channels, okay? They are not formed by leaky channels. So now we talked about how we sort of have this basal level of passing of ions in and out of the cell, right? That is affecting the potential energy across the cell membrane, right? So we're always having sort of an influx of positive and negative. Okay, so going back and forth between a little bit more positive, a little bit more negative. Well, that is not enough to actually form a graded action potential. Okay, so that is an important concept, that it is not formed by a leaky channel. Okay, is not done passively, we'll also say. And these are primarily, though, produced through chemically gated sodium channels. And we can even then further say that most of the graded potentials that are coming in to the cell body, okay, from other neurons, right? These are stimulated through chemically gated ion channels. Okay, so acetylcholine, for instance, we talked about, right? Neurotransmitters. So neurotransmitters signaling to this cell body to propagate another action potential. Well, some of these are all coming in at like 0.5, right? 0.25, right? 0.002, right? So all these would be graded potential, or graded potentials, but they were just small, and they sort of like diffuse out once they hit the membrane. They don't generate enough to come to the axon hillock point right here. Next, we'll say that that the graded potentials can also involve either the depolarization phase, right? Or it can involve the hyperpolarization phase. Okay, so in a repolarization phase, but you're hyperpolarized. And so what we can, and so to sort of explain that, we'll say that you can either have ions, and it's coupled with the type of ion that is being exchanged, right? So if you're bringing in positive sodium ions, right, then we're talking about bringing positive concentration inside the cell. We're becoming more positive, so then we will be moving more towards the positive end, right? But if we're bringing in negative ions, then we're bringing negative ions in, right? Or we're maybe exchanging positive ions back out of the cell, right? Well, then we're going to be going more negative. And so then the sort of the state of whether you're in depolarization, repolarization, or hyperpolarized, then that is dependent on the type of chemical ion that you are moving at the times, and sort of the channels that are open. Okay, so that's what I mean by when I say the graded potentials can involve either the depolarization or the repolarization or hyperpolarization of a membrane. And then we could also say another thing is that the stronger the stimulus, okay, the stronger the stimulus, right, the greater the change of membrane potential. And that just makes sense, right? So I sort of explained all that. If we have a 15 millivolt change from our resting membrane, then that'll give us enough to get to the threshold and kick off an action potential. But if we don't, if we have these small graded potentials, right, then those are going to sort of diffuse out, and they're not going to be enough to get us to that stimulus. So that's what I mean. Okay. The next part here I wanted to talk about 4.5. Okay, and this is propagation. Okay, propagation, right? And so what we'll say then is that action potentials are sort of these recurring events, okay, and they're basically happening over and over and over, okay. And what we mean by that too is that you know, across this axon here, okay, it's your axon hillock here, this point. So this is where an action potential is propagated from, and then it will, the action potential will just sort of carry itself across this axon, right, in a wave of depolarization. That depolarization will continue all the way across that axon until it gets to the axon terminal, and then it'll sort of transmit that signal, that electrical current across its synapse is what we call it. We'll talk more about that coming up. So we have this recurring event that's happening like a wave, right? And what we can say too is that it can either be continuous. Okay, so it can either be continuous, okay, or it can be, let's do one here, or it can be saltatory. Okay, so it can be continuous or it can be saltatory. And when we have continuous propagation, okay, we're referencing unmyelinated axons, okay? And we're going to say that these are slow, okay? And so what I mean is that this axon is not myelinated, okay? And so you just have the sort of the axon itself, right? And then you have that axonema around it, right? So just the axon itself and the axonema. And what we say is that the propagation of that action potential down the axon just runs across this axonema and the axon, okay? And we talked about before, okay, we talked about sort of this generic looking, right, axon. We talked about how there was these nodes, right? We call them nodes of Ranvier, right? And we talked about, let's do another color here. Let's just say, let's talk about, yeah, let's use yellow. So we have these myelin Schwann cells that are myelinating, right, across the axon, right? Well, you can either have a myelinated, which saltatory is myelinated, and we say that this is fast, okay? Or you can have a continuous propagation on an unmyelinated axon, which we say is slow. And to explain this a little bit better, so for the unmyelinated, we have within these segments here, we have these things called internodes. Okay, right? So these are internodal. It's within the axon. Okay, between the nodes, internode, right? And continuous propagation means that once we start our action potential wave and it's going this way, it will go continuously all the way through the internodes, okay? And it'll hit each one and go across the distance of each one of these internode sections, okay? Continuous propagation, all right? And that is very slow, okay, because it has to go all this distance. But we have something that is called saltatory conduction, okay, and this is due to the myelination of so just using this image here, you know, we have sort of the Schwann cell, right? And the Schwann cell is surrounding, and so we've got like layers, onion skin layers, right? So Schwann cell here, and then we have all this myelination, right? And so this axon is wrapped around, it is myelinated, okay? And one thing you need to understand is that myelin, okay, myelin itself pretty much blocks the flow of ions across it, okay? So if you're blocking the flow of ions across it in all these sections, right? And if you look at the majority of the distance actually between the axon hillock and the beginning of the axon terminal, majority of the distance here, okay, if you remove all of these yellow parts out of here, all you have left, okay, all you have left are these internodal areas, right? And the difference of these nodes of Ranvier, right? So you take these three together, right? So maybe the sum of those distance together is maybe, we'll say like this distance, right? These three together. But then if we were to take these four myelinated portions together, would say that maybe they are 1, 2, 3, 4. So maybe if you stack them side by side, right, you would have, so you see the difference, right? So the difference of the distance between these two, right, is pretty significant, right? So this is the difference right here. This point right here to this point right here. This is the difference of the distance. So what's happening is that if you have a continuous across an, or if it's an unmyelinated axon, okay, it is going to run continuously all the way across, has to hit every little piece of the internodes, and it's very slow. It takes time to go that whole distance, right? But if you have saltatory conduction, you have the ability, okay, to skip, to basically propagate this signal and jump each one of these segments of myelination, okay? And so you end up covering this distance very quickly, okay? So it's fast, okay? And saltatory, the term actually saltatory or saltaire means to leap, okay? So you get this leaping, and it's from the node of Ranvier to the node of Ranvier, okay? And if you look a little bit more in depth about these nodes of Ranvier, okay, we would say that it has a really high concentration, okay? This node of Ranvier here, right, has a really high concentration of sodium and potassium ion gated channels. Okay, and we mentioned that a little bit before as well. So we alluded to it, right? So if you go into these areas, you have a much higher concentration of these sodium potassium ion gated channels, which give us the ability to sort of conduct this action potential, right? And so it's a really fast and efficient way to transmit that action potential, okay, across the whole axon and do it quickly. And that's through saltatory conduction. Okay. Next, to sort of close this off, we'll go through this a little bit quicker. Okay. Or what I might do is, because this video is a little bit longer, I'm going to go ahead and we'll just stop right here, and we will pick up on 4.6 and talk about the synapses. Okay, so that'll conclude the discussion of action potentials. Okay, and sort of all the high yield information that I want you guys to know. Okay, about sort of initiating these action potentials and characterizing all the components, and then sort of the propagation of these across its axon. Okay, so thank you.